Hi students, hope you all are doing well. Today we're going to dive right into the world of enzyme inhibitors. These are the fascinating little molecules that act as master regulators inside every living cell. They're responsible for fine-tuning, slowing down, and sometimes even completely stopping the most critical chemical reactions in our bodies. So here's our lesson plan for today. First, we'll get a handle on what inhibition actually is. Then we'll meet two key types. One we'll call the imposter, and another we'll call the saboteur. After that, we'll see how our own cells use these inhibitors for some really clever self-regulation, before we wrap it all up with a quick summary. All right, let's get started. What exactly is an enzyme inhibitor? And what is its job inside a cell? Okay, so think of an enzyme as a tiny biological machine. It's constantly working, right? Its whole job is to convert one molecule into another. Well, an inhibitor is basically like a wrench in the works. It gets in the way and it can act as a brake on that machine, or even as a complete off switch. So, what's the deal with these things? Are they the microscopic villains of the story, or are they the essential heroes? Well, the truth is, it's a bit of both. Some inhibitors are, in fact, dangerous poisons, but many of them are absolutely essential for life. They're the regulators that keep our cells from just spiraling into chaos.
Here are the core details regarding this topic. Review them intently for their deemed crucial for your finals.
All right, let's get into our first type of inhibitor. I like to call this one the imposter, and you'll see why. It's a classic case of mistaken identity, but at the molecular level. Just take a look here. The competitive inhibitor is basically a molecular mimic. See how its shape is so similar to the real deal, the actual substrate? That similarity lets it slip right into the enzyme's active site. And when it does that, it physically blocks the molecule that's supposed to go there. It's kind of like someone stealing your reserved parking spot just because their car is a similar size. And this brings us to the key word here, competition. It's really just a numbers game. If you can flood the system with a real substrate, you seriously increase the odds that the substrate, not the inhibitor, is going to grab an empty active site. And what that means is this type of inhibition is reversible. You can actually overcome it just by adding more of the real substrate. Now, this isn't just some abstract concept. It has real-world life or death applications. Take antifreeze poisoning, for example. If someone ingests it, an enzyme in their body starts turning it into a deadly toxin. So what's the treatment? You might be surprised. It's a large dose of ethanol. Yep, the alcohol in drinks. The ethanol molecule acts as a competitive inhibitor, fighting for the exact same active site on that enzyme. By outcompeting the antifreeze, it buys the body precious time to safely get rid of the poison. It's literally a medical race against time, and it's won by an imposter molecule.
These are the vital facts linked to this section. Study them thoroughly as they are vital for examination point of view.
Okay, so that's the imposter. Now let's move on to our next character, which paints a very different picture. This one is less of an imposter and, well, more of a saboteur. See, this saboteur inhibitor plays by a whole different set of rules. It doesn't even bother with the active site. Instead, it attaches to a completely different part of the enzyme. But here's the sneaky part. When it binds there, it triggers a change in the enzyme's overall shape. It distorts the active site, making it useless. The key no longer fits the lock because the saboteur has warped the shape of the lock itself. And here is the absolutely crucial difference. Unlike with competitive inhibition, you can't overcome this just by adding more substrate. It doesn't matter how many substrate molecules are lined up waiting for their turn. The active site itself is damaged. The enzyme is pretty much out of commission until that inhibitor decides to leave. So let's put these two side by side to really nail down the difference. With competitive, just remember, it has a similar shape, it binds to the active site, and you can reverse it by adding more substrate. For non-competitive, it's the opposite. It has a different shape, it binds somewhere else, and adding more substrate does absolutely nothing. That's the core distinction. Okay, you might be thinking this saboteur-style inhibition sounds pretty disruptive. So why on earth is it actually essential for life? Well, let's take a look at how our own cells use it for some really elegant self-regulation. What we're looking at here is called a metabolic pathway, which you can basically think of as a cellular assembly line. A starting molecule gets converted through a whole series of steps, and each step has its own specialized enzyme. Now, in a brilliant process called end product inhibition, the final product of this whole assembly line acts as a non-competitive inhibitor for the very first enzyme in the chain.
This creates this beautiful, elegant feedback loop. You see, as the final product starts to build up, it shuts down its own production line by jamming up that first enzyme. It's the cell's way of saying, okay, we have enough of this stuff, let's not waste any more energy making it. Then, as the cell uses up the product, the inhibitor detaches and the whole assembly line just springs back into action. It's a perfect, self-regulating system.
All right, let's pull all this together with a quick summary to make sure these key concepts are locked in.
So this table really boils it all down for us. It lays out the two main types of reversible inhibition we've talked about. On one side, you have competitive inhibitors that fight directly for that prime real estate, the active site. On the other, you've got the non-competitive ones that sabotage the enzyme from a different location. And the biggest takeaway is how they respond to more substrate. One can be overcome, and the other simply cannot. So you see, from life-saving medicines to the very metabolic processes that keep us alive, inhibitors are the molecular switches that control biology. And that really opens up a fascinating question for the future, doesn't it? How can we harness this power to engineer brand new inhibitors that can fight diseases or even improve industrial processes? It's definitely something to think about. Thank you all for your attention.
Important terminology for this topic is shown on the screen, arranged according to various exam boards. These terms demand focused attention during revision as they are highly significant and must never be overlooked. Furthermore, a past paper evaluation is included for each board and paper format. Give this paper attention too, since it will strongly enhance and refine your preparation. Additionally, aims to grasp the meaning and usage of every term instead of rote learning them individually. Consistent practice of these terms in past paper questions will greatly boost your precision and overall exam results.
Please subscribe our channel to get more educational videos. Thank you.